To make the cake baking more effective, we add the required quantity of baking soda. If we want to sort onion faster, we add a small pinch of salt. Here, baking soda and salt speeds up the process. In this video, we will be learning about enzymes. So let us first try to define enzymes. For every reaction, there are two types of changes. They are physical changes and chemical changes. To boost the process and generate products at a faster rate, we add some extra substances to this reaction. So before heading into the core part of the topic, we should get familiarized with a few terminologies. How can we define a reaction? If any change has been observed in a chemical or metabolic conversion, then we can term them as a reaction. Another term is the substrate. The substance or the chemical which is converted into products is known as substrate. The next term is active sites. It is the region of an enzyme that binds to a protein or another substance during a reaction. These enzymes convert the substrate into the products. Now, let us study the definition of an enzyme. An enzyme is a biological catalyst or a protein catalyst which speeds up the rate of a specific reaction in the living cell. Enzymes are metabolic regulators and are highly selective in nature. What do enzymes look like? They are basically tertiary proteins with an active site, that is a protein part, and a cofactor, which is a non-protein part. The structure of the enzymes remain unaltered during the entire process. Here is one question for you. Do you know the known number of enzymes discovered so far? Any guess? There are more than 2000 enzymes known. Now it's time to check the main characteristics of the enzyme. First and foremost, there are two types: simple and conjugated enzymes. Simple enzymes are composed of proteins. Conjugated protein enzymes or the hollow enzymes are the enzymes that include a non-protein group which is tightly bound to a protein group. Most of the enzymes are water soluble. They have a three-dimensional structure with a substrate binding site. Let us now focus on the classification of enzymes. So they are classified on the basis of the type of reaction and on the basis of the substrate used. Based on the type of reactions, we can classify enzymes into oxidoreductases or dehydrogenases, transferases, hydrosylases, lyases, isomerases, and ligases. And on the basis of substrates, enzymes can be classified as proteases, lipases, carbohydrases. In this video, we will be checking on the different classes of enzymes categorized based on the type of reactions. First is the oxidoreductases. These enzymes bring about oxidation and reduction reactions, and hence they are known as oxidoreductases. Here, hydride ions of hydrogen atom are transferred during the reaction. These enzymes act as hydrogen donors when the substrate is being oxidized and are called dehydrogenases or reductases. Transferases are the enzymes responsible for transferring functional groups from one molecule to another. Considering the type of chemical group it transfers, transferases are of different types. Recall that in the priming step of glycolysis, we can see that the enzyme hexokinase, which belongs to the class transferases, phosphorylates alpha-D-glucose at the 6-carbon position by ATP to yield alpha-D-glucose 6-phosphate. Next is the hydrolases enzyme. The enzyme catalyzes reaction incorporating the hydrolysis processes. Hydrolysis of esters, ethers, peptides, glycosides, carbon-carbon, carbon halide and phosphorus-nitrogen bonds is done by this enzyme group. They break single bonds with the addition of water. Next, we will discuss on ligases. These enzymes perform the opposite function of hydrolases. The hydrolases add water to break bonds, whereas ligases remove the water component to join these bonds. For example, these enzymes catalyze the joining of carbon-oxygen, carbon sulfide, carbon-nitrogen and phosphorus-oxygen bonds. Liases are a class of enzymes that catalyze the breakdown of chemical bonds through methods other than hydrolysis or oxidation, leaving a double bond. Isomerases are a category of the enzyme that catalyzes reactions involving a molecule's structural rearrangement. That is, the interconversion of optical, geometric or positional isomers. Now, before learning more about cofactors, we should be thorough with the enzymatic action. For every biochemical reaction, energy is required, right? Let us understand the components of an enzyme and the catalytic cycle of the enzymatic action now. An enzyme marked as E has a substrate binding site which is marked as S. Recall that substrates are the reactants of a reaction. Once a substrate binds to the binding site, a highly reactive enzyme-substrate complex is formed and it alters its shape. One main feature of this complex is that it is short-lived. Here you can see that the enzyme breaks the chemical bond of the substrate and a new enzyme product complex is formed. Finally, the complex dissociates from its products marked in P. As with the other product, the enzyme is released. The enzyme-substrate interaction is described using the lock and key hypothesis and induced fit model theory. Let's now see what is the role of activation energy in reactions. The least amount of energy required to activate atoms or molecules to undergo a chemical reaction is referred to as activation energy. In other words, activation energy is the difference between the threshold energy and the reactant molecule's average energy. Let us now check the factors on which they are dependent. The activation energy is determined by the nature of the reactants and the effect of the catalyst used. This was about the protein part of the enzyme. Let us now learn more about the cofactors, which are the known protein component of the enzymes. Recall that hollow enzymes are the enzymes that include a known protein group tightly bound to a protein group. The non-protein constituents are known as the cofactors, which binds to the enzyme to make it catalytically active. And the protein part is known as apoenzyme. Cofactors can be metal ions, organic substances or other molecules that have the beneficial properties that are not found in amino acids. While the body can produce some cofactors such as ATP, others must be obtained only through food. Some examples include zinc ions, copper ions, potassium ions, etc. There are organic and inorganic cofactors as well. So we shall focus on the different forms of cofactors now. The first kind is the prosthetic groups. They are the organic compounds and are tightly bound to the apoenzyme. Hemoglobin molecule contains a heme, which is an ion which is bound to the protein part. And this heme enables the hemoglobin, that is the Hb, to carry oxygen. Flavins and biotin are other examples of the prosthetic groups. Next, we shall study coenzymes. They are also known as the small organic compounds, but their association with the apoenzyme is only temporary, usually occurring during catalysis. They are low molecular weight substances. The coenzyme usually acts as an acceptor or donor of an atom or a group. Most of these coenzymes are vitamins, vitamin derivatives or forms of other nucleotides. Two of the most important and widespread vitamin-derived coenzymes are nicotinamide adenine dinucleotide NAD and coenzyme A. A number of enzymes require metal ions for activity, forming coordination bonds with side chains at the active site while forming one or more coordination bonds with the substrate. For example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Have you ever thought, what if this cofactor is removed from the enzyme? The answer is very simple. The enzyme will lose its catalytic activity. It's time to check the various factors that are responsible for enzymatic activity. Have you ever wondered why milk is refrigerated? To prevent milk from curding, right? Refrigeration ceases the enzyme reactions and hence preventing curdling. By this example, what do you understand? The enzyme is inactive at very low temperatures, and since heat denatures protein, enzymes are destroyed at extremely high temperatures. Enzymes generally function in a narrow range of temperatures. Here, let's understand the meaning of optimum temperature. The optimum temperature is a range of temperature at which an enzyme shows its highest activity. The case is also the same for pH levels. Enzymes are extremely sensitive to changes in pH. Enzymes function in a narrow range of pH known as optimum pH. Now, let's see what happens when this enzyme or substrate concentration in the reaction medium fluctuates. First, let us check the enzyme concentration. The rate of reaction increases as enzyme concentration increases. After a certain point, enzyme concentration exceeds what is required. As a result, no enzyme-substrate complex is formed. As a result, the rate does not increase. If we check the substrate concentration also, the rate of reaction increases as substrate concentration increases. After a certain point, the rate does not increase because the substrate keep on decreasing as the reaction proceeds further. Next, we will learn the actions of inhibitors. The actions of inhibitors in enzymatic reactions are another factor which affects the enzymatic action. Enzymes catalyze almost every cellular processes. Have you ever thought of any way to alter this catalytic activity of enzymes? Here is the answer. Modifiers are the inorganic and the organic molecules that can change the catalytic activity of certain enzymes. They are of two types, activators and inhibitors. Those molecules which increases the enzymatic activity are known as activators, which are known as positive modifiers, while those that decrease the enzyme activity are known as inhibitors or the negative modifiers. Enzyme inhibitors are compounds that convert enzymes into inactive substances and thus reduce the rate of an enzyme catalyzed reaction. This is referred to as enzyme inhibition. So there are two types of enzyme inhibition that are widely recognized. Depending on the dissociation rate of the enzyme inhibition complex, there are reversible and irreversible inhibitors. Then, what is meant by competitive inhibitor? A competitive inhibitor is that has a molecule structure that closely resembles the substrate and inhibits the enzyme's activity. Since it has close structural similarity to the substrate, the inhibitors compete with it for the enzyme's substrate binding site. As a result, the substrate cannot bind and the enzyme action is reduced. In the inhibition of succinic dehydrogenase by malonate, which is structurally similar to the substrate succinate. These competitive inhibitors are frequently used to control bacterial pathogens. Do you know that ribozymes, that is the ribonucleic acid enzymes, are the nucleic acids with a catalytic power? They form a covalent bond between molecules. Hammerhead ribozyme is an example of a ribozyme. Sometimes these ribozymes often cause diseases also. Like the ribosome hepatitis delta virus is a human pathogen causing delta hepatitis or hepatitis D. Now it's time to wrap up the session. Let us summarize what we have learnt so far.
Manorama Horizon. Learning made simple.